Have you ever wondered how scientists see things that are too small for our eyes? Today, we are entering the microscopic world, a world of cells, microscopes, and amazing specialization that makes plants and animals survive. By the end of this lesson, my viewers will be able to discuss the cell structure, prepare slides for observations, estimate the size of the cell, state the differences between light and electron microscope, draw and label both animal and plant cells as seen under the two microscopes, outline the adaptations of the parts of the cells, that is animal and plant cells, prepare temporary slides for observations and estimation of cell size. Appreciate the lesson by sharing, liking, and subscribing for more online classes. Welcome to the cell structure and specialization. Let us zoom in. Preparation of temporary slides using light microscope. Materials we need microscope, glass lines and a cover slip, water, specimen that is onion peel or chick cells. Finally, we need iodine or methylene blue stain. Steps followed to prepare a temporary slide. 1. Place a drop of water on the slide. Add specimen carefully. And 1 drop of stain, that is we need either iodine or methylene blue stain, and 1 drop. Lower cover slip gently to avoid air bubbles. Observe under low power to high power. Estimation of cell size during microscopy. Formula: cell size is equal to diameter of field of view divided by number of cells across. I repeat the formula for estimating the cell size during microscopy. Cell size is equal to diameter of the field of view divided by number of cells across. Steps followed to calculate the cell size. 1. Measure field of view under low power. 2. Count number of cells across. 3. Calculate approximate size of the cell. This one is used when micrometer is unavailable. Differences between light microscope versus electron microscope. Light microscope uses light to operate, while electron microscope uses electron beam. In terms of magnification, light microscope can magnify an object up to 1,000 times, while electron microscope can magnify an object up to 1,000,000 times. So it's very high in terms of magnification. In terms of resolution, light microscope is very low, while electron microscope is very high. Living cells. Light microscope, yes. Electron microscope, no. Image, light microscope, color, then electron microscope, black and white. NB, electron microscope shows finer details compared to light microscope. Cell structure. Electron microscope view. Plant cells structure and function as seen under electron microscope. Under electron microscope, we can be able to see the following: cell wall that supports and protects the cell, chloroplast that performs the process of photosynthesis, vacuole acting as a storage tangidity of the cell, nucleus controlling all activities of the cell, animal cell as seen under electron microscope, cell membrane controlling movement of substances in and out of the cell, mitochondria production of energy, ribosomes protein synthesis, lysosomes helps in digestion. Remember this: animal cells lack cell wall and chloroplasts. Differences between plants and animal cells. Differences between plants and animal cells. Plant cell, animal cell. Plant cell wall present, animal cell absent of cell wall. Plant cell have chloroplast, but animal cells have no chloroplasts. Plant cell regular in shape, but animal cell irregular in shape. Plant cell have large vacuole, but animal cell have small vacuoles or maybe none. Specialized cells and adaptations. Specialized cells and adaptations. Plant cells. Root hair cell, long projection for absorption, for water absorption. Silent vessels, thick walls for water transport. Palisand cells, many chloroplasts for process of photosynthesis. Animal cells. Animal cells have red blood cells which have no nucleus. Then because there's no nucleus, this makes them to carry more oxygen because of hemoglobin. Nerve cells, long axon for rapid impulse transmission. Sperm cell, tail for movements. Remember, structure is always related to function. Assessment questions and answers. Which part of the microscope controls the amount of light passing through the specimen? Eyepiece, objective lens, diaphragm, stage. The answer is C. Question 2. Which microscope can be used to observe living cells? Electron microscope, scanning electron microscope, transmission electron microscope, light microscope. The answer is D, light microscope. Question 3. The main advantage of an electron microscope over a light microscope is it is portable, use of color images, higher resolution, ability to observe living cells. The answer is C, higher resolution. Question 4. What is the correct order when focusing a specimen and a light microscope? High power, low power, low power to high power, medium power to high power to medium power. The answer is B, low power to high power. This is the correct order when focusing a specimen and a light microscope. You start from low to high. Question 5. Section B. Cell size can be estimated by dividing the diameter of the field of view by total magnification, by number of cells across, by the size of the eyepiece, by resolution of the microscope. The answer is B, by number of cells across. Question 6. Why is estimation of cell size important in microscopy? Why is estimation of cell size important in microscopy? To increase magnification, to identify stains, to know approximate size of cells, to focus the microscope. The answer is C, to know approximate size of the cells. Section C, question 7. Which structure controls all activities of the cell? Mitochondrion, nucleus, ribosomes, vacuole. Mitochondrion is for energy, nucleus to control the activities of the cell, ribosomes to synthesize protein, and etc. So our answer became B. Nucleus controls all activities of the cell. Question 8. Which organelle is responsible for energy production in cell? Ribosomes, lysosomes, mitochondrion, nucleus. The answer is C, mitochondrion. Question 9. Which structure is found in plant cells but not in animal cells? Cell membrane, nucleus, mitochondrion, cell wall. The answer is D, cell wall. Cell membrane present in both, nucleus present in both, mitochondrion present in both, but cell wall absent in animal cell. Question 10. The function of ribosomes is to store food, digest waste, synthesize proteins, control cell division. The answer is C, synthesize protein. Section D. Plant versus animal cells. Which of the following structures is present in plant cells only? Lysosomes, chloroplast, ribosomes, cell membrane. The answer is B. Chloroplast is only present in plant cells. Question 12. One major difference between plant and animal cells is that plant cells have mitochondria, lack a nucleus, have a large permanent vacuole, are smaller in size. One major difference between plant and animal cells is that plant cells have mitochondria. No, they both have. Lack a nucleus? No, they both have. Have a large permanent vacuole? Yes. Are smaller in size? No, animal cells are smaller in size. So question 12, the answer was C, have large permanent vacuole. Section E, specialized cells. Which feature enables red blood cells to transport more oxygen? Presence of nucleus, biconcave shape, presence of chloroplasts, weak cell wall. The answer is B, concave shape. Question 14. Root hair cells are adapted for absorption because they have thick walls, have many nuclei, have a long projection, contain chloroplasts. The answer is C, have a long projection. Question 15. Which specialized cell is responsible for rapid transmission of impulses? Muscle cell, red blood cells, nerve cell, sperm cell. The answer is C, nerve cell. We have learned how to prepare lines, estimate cell size, compare microscopes, and explain how cell structures are adapted to their function. Remember, cells may be very small, but their roles are massive. If you found this lesson helpful, why don't you like, subscribe, and share? In the next lesson, we'll continue exploring more about biology. Stay curious and turn on your notification. Bye.